Yeah. Okay. So thanks for having me. It's it's a real pleasure for me to be here. Never been in South Africa. I'm very new to data collection, so I've learned so much over the past 2 days. So what I'm trying to share with you is maybe some considerations for data analysis. So some of the content will be a bit general, but I'll try to channel it a little bit for things that may be useful for projects, for Depgen projects. I guess the ones in the next round, but maybe because you mentioned Depgen 2, maybe that could feed into a follow-up project. So just wanting to introduce myself. So I'm started my group at the University of Queensland at the end of 2020, and essentially, let's keep this slide here. We're about 6, but we're really interested in understanding the genetic causes of phenotypic variation. And so this is very general. This is our sort of overarching question. And that gets manifested in different questions, specific questions about the genetics of conditions like diabetes, but also complex traits like body mass index, etc. So I guess the guiding principle of our research, and this is probably relevant for this meeting, is that we're trying to come up with statistical methods. So this is what we develop in the lab. We develop statistical methods to better analyze genomic data. And the guiding principle is really to be able to extract as much information as possible from the data. So I guess it fits with this context where we have a data generation which it's known from the start that statistical power will be limited. And so everything we can bring in terms of new ideas for analyzing data to extract as much information as possible, I believe will benefit this project. So I guess we're all here because we recognize that the majority of our global community has been underserved by genetic studies. This is a statistics that came from a paper published in 2019, which showed that there is an overrepresentation of European ancestries in current genetic studies, mostly in GWAS. And I would like to thank Vivian and Jared yesterday who gave a very nice introduction for me, so I don't need to go back here to explain that again. But unfortunately, this overrepresentation has implications. And one of them is that our ability to predict risk of disease, for example, just to name a few of those implications, is severely reduced in overpopulations. And so we're talking between 50% and 75% loss in prediction accuracy. 75% is probably the number we want to keep in mind because that applies to African ancestry populations. So what exactly are we missing to bridge the gap? So I guess the number one thing is data, and I think this particular initiative is addressing it upfront, generating data for those underrepresented populations. But I would argue that we also need a better understanding of this phenomenon. We observe this loss of prediction accuracy, this lack of cross-variability, but what exactly is driving this? And I believe the more we understand that phenomenon, the better we can extract information. And this is where some analytical tools can help to reduce the gap as well. So my presentation will have 2 main parts. So the first one, I'll try to share with you some of our understanding of that phenomenon of loss of prediction accuracy from somewhat theoretical point of view, but I will also show some empirical analysis. So this relates to papers we published a couple of years ago. In the second part of the presentation, I will focus on some analytical tools or some ways we can think about analyzing data from underrepresented populations to yield as much power as possible. So if we think about this phenomenon of lack of transferability, it is consistent with a lot of potential explanation. The first one is that we have ancestry-specific causal variants. And I would argue that this is a bit of it may be the worst case scenario, because if that's true, it means that the only way we have to reduce the gap is to generate data from those underrepresented populations. But in some cases, if we think one of the lessons we've learned from knowledge of GWAS is that in order to make discoveries, we will need very large sample sizes. And for some part of the world, it may not be relevant for the African continent, but in the context of Australia, for example, when we think about some of our indigenous populations, even if we could genotype and sequence all of them, we may not have large enough sample sizes. So that's why I believe it could be, if that's the reason, that could be a worst case scenario. But there could be other explanations. Another explanation could be, well, the causal variance, the sort of underlying relationship between variation in DNA sequence and the risk of disease is the same across populations, but it's just that the effect of those variants will vary. And I believe this is a more practical situation because we can still try to learn from this, identifying those causal effects, and maybe we just need to characterize how the effect of those variants will impact populations, which is probably more trial. And there is another, probably it's a bit of a theoretical explanation, but this is a context that's relevant for what I'm going to show next, is that we can imagine that the causal variants are the same, that the effect sizes are the same, that even the heritability of those conditions and traits are the same, but we just have frequency differences in our genetic variants. And this is something we observe. Certain genetic variants are more or less frequent in certain part of the world, but is this going to be enough to explain the loss of prediction accuracy? And this is where we started the study. So we started the study by essentially recognizing that, because of demographic relationship between human populations, we do have those differences in allele frequencies and linkage disequilibrium. So certain blocks of DNA that are transmitted from one generation to the next, well, it's not necessarily the same blocks of genetic information that are transmitted, because their frequency can be different between populations. So this is something we know, this is something we observe. But what we really wanted to understand in that study was to try to quantify what if that's the only factor, how much would we expect of essentially losing prediction accuracy. And I'll come back to that at the end, because if we understand that, it has some implications about how we can solve it. So this is exactly what I just said, how to quantify the loss of prediction accuracy that is only attributable by the sort of allele frequencies and L differences between populations. So here we're assuming that everything is the same, causal variants are the same, effect sizes are the same, everything is the same, it's just that we have those differences that are because of demographic history. And so this paper was published in 2020, so if you want to have a look at more specifics of the paper, we can come back to that. But in this paper, we essentially made two assumptions. The first one is well, we wanted to have some theoretical framework, because I was trained as a statistician, so I can't publish a paper without a bunch of equations, so we had to generate some of those. But we had this model where we say, okay, well, actually, even if we are making all those assumptions, simplifying assumptions, we wanted to at least have a theoretical framework that would allow us to see where things like differences in effect sizes or differences in heritability between populations may play in that loss of prediction accuracy. So we integrated that in the model, so we assume that the causal variants are the same, but the effect sizes are correlated with a correlation which is not exactly one, is captured by this parameter. And the second assumption we've made is that we have a reference population, just to mirror what we have now in European populations, where we've conducted the large GWAS and we have a bunch of hits from that populations. And we know how much they explain in that reference populations. So in the context of depression, that will be, we have a large GWAS of depression out there conducted in European populations, and we know exactly how much of polygenic scores from that large GWAS explains in European populations. So I'm going to spare you the expanded version of that equation, but what we model is essentially the ratio of prediction accuracy. So here, if we think about population 2 being African populations, we wanted to see what is the relative accuracy of our polygenic score. If we look at the accuracy R square in population 2 divided by the R square in population 1, how can we model that? So essentially, we saw that it was a function of many things, but we can group those things together. So we have a bunch of parameters that are capturing the environment. So this is essentially all the reasons why the effect sizes may not be different, so that may be different between populations. So that could be because of G by E interactions, potentially G by G as well, and differences of heritability. But this is something we had in the model. We said we're not going to look at that first. And then we had a bunch of other set of parameters, which were essentially the allele frequencies at the SNPs that we have in our polygeny score. So if we say we derive a polygeny score from, I think it was 190 associations from some of the papers discussed yesterday. So if we take those 190 variants and we want to predict depression, say in African populations, what I mean here, those PK1 and 2 are essentially what is the allele frequency of one of those 190 variants in European and the allele frequency in African populations. And we have another set of parameters there, which it's not necessarily something we can observe, but this is essentially saying that, okay, we don't know the causal variance. This is something that we may not have observed because of lack of repetition accuracy, or maybe the causal variation is just more complex. Maybe it's a copy number variant that we're trying to capture with our SNP data. Whatever that causal variant is, what seemed to play there is how those SNPs that we're using in the polygeny score, how they are correlated, they are tagging those underlying causal variants. And that can be different between populations. And so I'm trying to illustrate that here. So imagine this one here is a true causal variant. So just some sort of linear representation of the genome, and each of those circles will represent variant. So let's imagine that the causal variant is this one that is not observed, so I should be using the pointer, is not observed, but how the SNPs nearby, which may be the SNPs being written on the polygeny score, are correlated or tagging that underlying causal variants will change between populations.
Anthony, can you mute your microphone? You've just joined us. Thank you. Sorry.
Thanks for coming. Yeah, we mute it.
Sorry.
No, it's all right. So this is a set of so we have this theory. So okay, we kind of see all the moving pieces. Again, we made assumptions, but we see there is a part that prediction accuracy that is coming from reasons why effect sizes may be different, why heritability may be different, and that involves sometimes the environment. We have a component there. We have things like differences in allele frequencies for the SNPs we're using in our polygeny scores, which can also differ between populations. And we have another component, which is how those SNPs that we're using in our scores are tagging those underlying causal variants. So that was all good in theory, but in practice, as I said, we may not know the causal variants. So we use this kind of heuristic thing, which came from a paper that some of my colleagues published in 2017, which kind of say that when you have a GWAS hit, if you look around, most likely with some high probability, the causal variance will be in variants nearby, within say 100 kb, with some correlation larger than 0.45. Those are very arbitrary numbers, but that came from that paper. But so essentially what we said is that even if you don't know the causal variance, you can just test all the variants around your GWAS hit and assume that what if this is the causal variance, and calculate those quantities and look at how they differ between populations. So let's now let me walk you through a simulation study that we implemented to see how this theoretical framework will work in practice. So we run the simulation in the UK Biobank. So in the UK Biobank, you have about 4 major ancestry groups. So the majority are European ancestries. We have about 7,000 African ancestries, which half of them are continental Africans. The other half are probably Caribbeans. We have about 2,000 East Asian and just about 9,000 South Asian. So we simulated a trait where under our assumptions where the causal variants are the same, everything is the same, etc. And we ran GWAS in the European populations, and we ran GWAS in the European populations. We identified a set of top SNPs for the simulated trait, and we went in our target populations and look at what is the relative accuracy, essentially the ratio between the prediction accuracy in the target sample versus the prediction accuracy in the European ancestry populations. And so we use 1,000 genomes for calculating those candidate causal variance relationship by assuming that we don't know the causal variant, but nonetheless, we can scan around the GWAS hit all the variants within 100 kb that fulfill our criteria and pretend they are the causal variants. So, yeah, these are just some of our simulation scenarios. I think by and large, we do see very consistent results. So we vary things like the number of causal variants, variability, but I'm not going to bore you with this. So this is the first set of results. So this is essentially a simulation setting where, as I said, we have the same causal variants. The effect sizes are the same. If we let's just focus on the gray box plots. So the gray box plots are tracking the relative prediction accuracy. And we can see that under these scenarios where everything is the same, if you look at the African ancestries, we essentially already losing. Essentially, we have a relative accuracy that is just above 40%. So it's still higher than what we have in real data, which is essentially about 25. So we have a 75% gap. Here we have about more than 50% gap, just because of those allele frequency differences between populations. So I think that was an interesting eye-opening. And the other bars are essentially our predictions. So if we look at, we might have the right columns displayed somewhere. But essentially, I remember well, the golden bars are essentially what we derive from our model and predict that relative accuracy. But this is a bit cheating because here we know the causal variant for real because we simulated that. And the blue is when we use this sort of heuristic strategy by pretending that maybe the causal variant is a variant nearby. And it was doing okay. It was kind of overestimating or maybe underestimating the role of genetic differences, but it was still consistent with a large prediction. And just briefly want to emphasize those green bars. So those green bars is what you would have if the GWAS hits were the causal variants. This is what you would expect. You would expect something here, and that's actually so far from what we observe. And just one of the reasons we kept it there, it was kind of educational because we kind of observed in the literature that once we started compiling enough evidence of that loss of prediction accuracy between populations, starting to see people speculating on the fact that, okay, this is strong evidence that the architecture of those traits are very different. But I think that was a shortcut. And this is a shortcut. It's a caveat that you can fall into if you think that your GWAS hits are causal variants, and I believe they're not. So this is something to keep in mind. And so the rest is just to say that across all the scenarios, we have very, very similar. So before moving on, one thing I just wanted to flag is, okay, this is all good, it's theory, we make assumptions. I wanted to challenge some of our assumptions just a little bit because we know for many of those traits that we have, they've been shaped by natural selections, selection pressures over thousands of years. And that essentially manifests in some sort of relationship between effect sizes and allele frequency. So we're trying to fiddle a little bit around what will be the impact of those selection pressure. And by and large, just skipping the details, the bottom line is our model, at least the way we try to predict the contribution of allele frequency differences and LD differences between populations is actually underestimated. This overestimates the genetic cause of loss of prediction. So it is a good model, but it's clearly not perfect. This is something just to say in passing. Okay, so but we still wanted to see, we have a framework. So just to remind you, the framework is, I have a GWAS in European populations. I have a bunch of SNPs, so like this is a bunch of SNPs, real traits we had in the UK biobank. Let's say we start with height. So for height, we have 1,000 variants associated with height in Europeans from just the UK biobank. Our framework was to say, if we just look at those 1,000 variants, we look at allele frequencies between Europe and other parts of the world, and we look at differences in local linkage disequilibrium between those lead variants and variants nearby and how that differs between populations. We should be able to predict how much our predictability score will drop just because of those factors. And if that's correct, it means that almost by subtraction, we can infer what will be the role of those other forces? Like, what will be the role of G by E? What will be the potential role of differences in heritability? And so we did that. So this is just focusing on those three populations. Probably can't see much, but I'll jump to the conclusion. And this is just illustrating what I was saying, is that we have the observed relative accuracy, we have a predicted relative accuracy, and we can look at the difference of the loss of prediction accuracy and how much it is explained by. I should have changed that slide, so it's a bit convoluted. So the conclusion, the main message here is that across those three populations, so each of those columns represent populations. So on the top, on the extreme left, A and D are South Asian populations, B and E are East Asian populations, and C and F are African populations. So we kind of order by genetic differentiation away from Europe. And so what we see here is that our relative accuracy is very similar to what was reported. At least this is the observed one. We have a relative accuracy of about 24% on average across traits. And using our model, we can attribute 86 or something around 80% of that loss just because of allele frequencies and allele differences between populations. And I think this is, again, we need to validate that in many ways. But if that's true, it means that we can potentially recover those 86% loss by having new methods that are accounting for LD and allele frequency differences better than what we have now. Because this is purely due to those factors, and this is something we can easily observe. So this is it has potential important implications there. So I was a bit hesitant here to claim how true that is, because we've made a lot of assumptions in that study, but I was happy a few years ago. That was last year, actually. Last year, this paper published in Nature Genetics, which we tried to do the same, address a similar question, but using a very interesting design. So essentially, their approach was to see what was the correlation. Let me put it this way. They were interested in knowing whether causal variants had the same effect sizes in European and African ancestry populations when they were observed in the same individual. So essentially, they focused on admixed individuals, so African American and probably some Caribbeans as well. And so in these admixed individuals, part of their genomes can be traced back to European ancestors, and other parts of the genome can be traced back to African ancestors. So, but in this design, the cool thing about it is that if you see at a particular position on the genome, if you can quantify how the allele that is coming from Europeans is affecting your trait versus how the allele on the same variant, how the allele that is coming from your African ancestor is affecting your trait, and if they are different, this is essentially, well, you can use this kind of information to learn about differences in allele frequency in LD. And so, by and large, what they did in that study, they found that causal variants seemed to, those effects were highly correlated. So if a variant increases your risk for disease, it doesn't really matter if it comes on a European haplotype or if it comes on an African haplotype. And I think that was interesting. So there are limitations of that study. One of them being that their sample size were small, and I'm working, many others are also working along those lines to try to get better estimates for this kind of model. But at least it went in the same direction, suggesting that causal variants of complex traits are probably one largely shared across populations. And secondly, their effect sizes may not be that different. So it doesn't rule out, it's not a perfect correlation. I think their meta-analyzed correlation is about 0.9. So it's not 100% correlated. So there is still room for things like G by E or G by G, etc. But it's probably not going to be the major determinant. And I think this is a good news in some ways, especially for initiatives like this one, because it means that we don't need to necessarily reach those large numbers. If we found good ways to sort of transfer the information, we can still achieve good discovery yield.
Can I ask you a question? Because this issue has been really the driving factor for doing genetic studies in Africa. So how do we reconcile that? The issue of equity we've been talking about, Africans underrepresented genetic studies and so on. So if these findings are transferable to across populations,
Yeah,
how do we reconcile that?
This is a great question, and I think the answer to your question comes from the fact that findings from GWAS are not causal. So we still need, if somehow finding from GWAS were causal, I guess we probably would not be in that position today. But yeah, it's exactly because they're not causal that we still need to get at the causal variant. So in my opinion, this is really where an initiative like this can fast track that discovery, because technically, if we have the causal variants, we may still have some loss of prediction accuracy because of differences in the environment. But it's not going to be, at least as far as I understand this question, it's not going to be as dramatic as what we observe now. So yeah, there is space, and typically bringing African ancestries because of smaller healthy blocks, etc., has a potential to accelerate discovery of causal variants. So, just to take home message here, so I have a couple of take home messages for that first part, and I hope I'm not speaking too slow. Please let me, I'm not tracking the time, so let me know. So the take home message is that the relative accuracy is lower than expected if the GWAS SNPs were causal variants, but I believe this is the wrong conclusion. We shouldn't be thinking about drawing biological conclusions from that loss of prediction accuracies because essentially because GWAS SNPs are not causal. So we have a method that can predict the relative accuracy, even when the causal variants are not known. I think this was a strength of that approach. But we can obviously think about improving that method if we bring sort of fine mapping tools into it. And this is something, a very quick comment I've made. Our method is still conservative because it doesn't really fully account for the genetic architecture, and especially for things that are shaped by natural selection. Yeah, I guess this fourth take-home message is a bit related to the first one, actually. So the statistical prediction of the role of habitat frequency differences may be misleading. Yeah, I guess this is, I forgot what I wanted to say. Actually. So this presentation is going to be on YouTube, right?
Yeah.
Sorry, YouTube. So I don't remember what it was for. So 5. Yes, so I think here just wanted to emphasize actually, yes, where are the knowledge gaps? So, yeah, I still don't remember what I wanted to say before, but for 5, so this strategy that we have, not just us and some of the papers that people have published around these questions lately, I often ignore the presence of G by E or G by G. And maybe some of those conclusions will be slightly distorted. Most of those methods, the papers are published and papers that others are published. We try to think about it as it's all additive. I think we need to fill a gap there to have some understanding of what if we know that for many complex traits, those non-additive effects are not contributing hugely, but even if they're contributing a little bit, how that could distort our conclusion regarding prediction analysis. I think this is a big gap that needs to be filled. I think there is another gap we need to fill as well. Is maybe we need to emancipate ourselves from this sort of categorical view of ancestry. Right? Because we think about African ancestry. I guess I'm probably preaching to the choir here. I think you all understand here the genetic diversity there is in this continent. So I just want to pause here. It's the first time I hear you talk in Africa, actually. So yeah, it's great. Thank you. So, but I think we have to think about, even for our polygenic scores, how to think about ways that, because nowadays you have methods that will tell you if you want to predict a trait, you can use this polygenic score if your person is European ancestry. If your person is African ancestry, use the other polygenic score, which is a bit better, etc. But it's still categorical. And I think what we need is something like more continuous. If someone has a gradient of different ancestries, how can you use that information to have the proper prediction? And so I think this is again a space where we need to bring more knowledge. So I'll probably be a bit faster on the second part because I just wanted to talk about two methods we can use now to accelerate or improve discovery of GWAS in a setting where sample size is limited. And so the first method is something we published last year, and it sort of came from this idea that we can use polygenic spores from other populations to boost the discovery of new GWAS in underrepresented populations. So let me walk you through the statistical intuition. So in a study of GWAS, you have your phenotype, you have the SNP you want to test, and you have everything else, your residuals. And so the way we think about increasing power is essentially by reducing the variance of those residuals. And so typically in standard epidemiology, we do that by minimizing differences between people that are not related to what we want to test. So we want to account for sex differences, we want to account for all the artifacts, year of birth, etc. But one thing we can do is that we can also account for the polygenic score for that particular trait, because even if, probably have that in the next slide, well, just before going there, the strategy is that essentially we test the phenotype and we have the polygenic score as a covariance. And so why this is principled to work is because even if we have this loss of prediction accuracy, it's not nothing. In some cases, we're losing 75%, but we still have 25% that we can expand. So in the context of depression, I think it was, talking very small numbers, I think on the Likely scale was something like 2%. So maybe in the context of depression, it's going to be tricky. I don't know the answer here. But I think so what we showed in that study was this is principled to work because you do expand. And so we tested that in East Asian populations with different traits, and this is just an example for height. And we showed that if you account for this polygenic score using the same data, not doing anything else, just bringing a polygenic score from another study, we can discover 200 more hits by doing that. So this is an outlier. It's height, it's highly heritable. But what was really interesting is to show that in East Asian populations, it was not consistent across all traits, but in some cases we were able to make additional discovery. Still very modest. But if I highlight, for example, I guess maybe for type 2 diabetes, we discovered 5 more associations by essentially, so we were running this GWAS in East Asian populations and bringing polygenic score derived from GWAS conducted in European populations as covariance. So I think this is something we can think about implementing in the context of DepGen, because, well, I guess I know there's some project about polygenic scores that are in the making, but that could also be part of the pipeline for GWAS. And so the second method is something that I actually added this morning, following some of our discussions yesterday around the generation Malawi. And I was thinking about a series of methods that have been developed. I haven't developed any of those methods myself, but they've been in the literature since around 2017. And those are methods where you can essentially borrow information from relatives, even if they're not genotyped, but you know their case status. And so what they showed in that first paper in 2017 was in the case of Alzheimer's. So they had a cohort which was relatively young, so they didn't have enough time for them to develop late onset disorders, but they have family history on those participants. And so that information, because you share 50% of our DNA with our parents, having that information allowed you to still run some sort of GWAS by proxy for those phenotypes you have measured. And more recently, this has been sort of more generalized in the context where you have a combination of people who are genotyped, cases that are genotyped, and cases that are not genotyped. And I think in the context of Malawi, probably other data sets in Ethiopia or Nigeria, we may have them as part of relative of our participants who have been phenotyped, So it doesn't necessarily impact the budget in terms of genotyping, but this is information that can be brought in the analysis to increase power. So that's the two things I had. Probably should I have like a recap slide, but I'll just maybe in a couple of words summarize what I just shared with you. So the first thing was really to share some understanding of the sort of lack of prediction accuracy, and we've got to understand now that this lack of prediction accuracy is largely driven by allele frequencies and health differences between populations, which means that there is a huge opportunity for us to resolve that gap if we can identify those. And the second part of the talk was mostly kind of examples about methods we can use, and there are more methods out there that, and I believe, because we are at the start of this project, thinking about implementing those kind of methods will be principal to make more discoveries. So